Καλό απόγευμα. Καλή χρονιά. I'll switch to English now. It is an honor to be here, invited by the Cyprus Biological Society, the Ministry of Health, the Medical School. Είναι τιμή μας που ερχόμαστε από την Αμερική και είμαστε εδώ. Και είναι πραγματικά χαρμόσυνο γεγονός να βλέπω ότι στην Κύπρο αρχίζουν να γίνονται τέτοιες εκδηλώσεις. Είναι πολύ χαρμόσυνο γεγονός και είναι τιμή μου που βρίσκομαι εδώ. Αλλά θα κάνω switch στα αγγλικά, γιατί έχω συνηθίσει να τα λέω αυτά στα αγγλικά. Και θα ξεκινήσω με τον τίτλο. Και όλους τους χορηγούς βέβαια, τους ευχαριστούμε. Λοιπόν, και απολογούμε ότι δεν είναι πολύ καλή εικόνα. Αλλά θα κάνω ό,τι μπορώ. So, today I will be talking about mapping EMT and therapy-resistant phenotypes in lung cancer tumors, liquid biopsies with single-cell proteomics. So today, there will be a few concepts that I'll talk about. I won't go into many details, but I hope I can help you realize the strengths of some approaches that I use in my lab that could be very translational and clinically applicable to study cancer at a very detailed level, at the single-cell proteomic level. So first I will talk a little bit about the technology we use in my lab, which is called cytometry by time of flight, and the strengths it has to study cancer. I will also talk about a tool that I developed during my postdoc that helps you analyze clinical specimens at the single-cell proteomic level. And I will talk about its applications for non-small cell lung cancer, but also how I bridged previous work to work in other types of lung cancer, like small cell lung cancer, and in general to study solid tumors, circulating tumor cells, and liquid biopsies. Okay, so whenever I give this talk, I do like to give to the audience the key terms and concepts. One of them is I will be talking about phenotypic plasticity and heterogeneity. Many people who study cancer, most of us focus on the genetics, but there's more and more studies now that show that other mechanisms drive cancer progression that are not necessarily driven by mutations. Cancer cells have the ability to change and adapt to stimulus very fast that would lead that cancer to be therapy resistant and progress fast. And those are the kind of things that we study in my lab, specifically epithelial to mesenchymal transition, which I'll talk a little bit about just to give you the background. I'll talk a little bit about how cells can visit states and change their states to confer them resistance or survival abilities when the patient is under treatment, right? And this is a very dynamic process which gives the cancer adaptability to whatever you throw at it. And then finally, again, what I'll be talking about is by using this very cool technology, you can study these kind of processes at a very detailed level, specifically, in my case, single-cell proteomic level. Okay, so what is mass cytometry, otherwise known as CyTOF? If you know about flow cytometry, you're halfway there. So instead of using fluorophore conjugated antibodies for flow cytometry, you use metal conjugated antibodies. So now this gives you the ability to not only study, let's say, up to 10 to 12 antibodies at a time, because flow cytometry has become better, because you avoid the spectral overlap of fluorophore antibodies and you use metal antibodies, you now have the ability to interrogate up to 50 antibodies per cell, sorry about that, in your clinical specimen. And theoretically, you can go up to 100 markers per cell. So this gives you now a problem because all of a sudden you can study all these processes that we all are interested in our labs and our research at the same time, right? So now the issue becomes, what is your question? What are you interrogating with all these capabilities? And then how do you actually get to the bottom of the data? Because now you have very high dimensional data that you need to understand, visualize, and use to treat the patient or understand the cancer. Sorry again. So I'll be talking a little bit about ways to understand this kind of data that you're getting from each patient. Okay, so like I said, in my lab, we're interested in this process called epithelial to mesenchymal transition, right? Now, this process was first described in developmental biology. It's a process through very defined epithelial cells that have very defined structures and functions undergo through different stimulus in the microenvironment a phenomenon where they dramatically change their morphology, their behavior, and they become mesenchymal, and now they're very migratory and therapy resistant. Now, the issue in the field was that it's not just this state and this state. There's many intermediate states that up to very recently had not been defined at the single cell proteomic level. Now, why is it important to do that? If you know every state's ability to metastasize or be resistant to therapy, then you have a way to target it. You first have to understand it, describe it, and then try to target it. And because it's such a complex and also reversible process at any given time, when I started this research, I wanted to use CyTOF to well-define with proteins that would describe each and every one of these states. And it's important to understand that it's also reversible. So for example, in lung cancer, EMT, this ability to change, has been linked to therapy resistance, tumor immune evasion, recurrence, and poor clinical outcome. But the thing is, it is believed that this takes place when cells detach from the primary tumor and start metastasizing in the blood. But then they have to reverse once they reach the new site and become again epithelial to grow a new tumor. So the plasticity phenomenon is very important for cancer. It's what keeps them going, right? So when I was doing this project, I used CyTOF, so I was able to interrogate all these proteins here at the same time. And I used markers that define EMT, survival, signaling, apoptosis to get a better feel of the characteristics of each and one of these states. So I induced EMT in lung cancer cell lines with treating with TGF-beta, and then I withdrew TGF-beta to induce the reverse, okay? So then having all this data, the high-dimensional single-cell proteomic data, my goal was to build a map that anyone could use to assess clinical specimens from lung cancer patients. So pretty much we built this reference map that depending where your cells of analysis fall, it will tell the clinician or the researcher, this cell is mesenchymal, this cell is epithelial or partial EMT. Okay, so just to show you a little bit of the in vitro biological data, and I know the pictures are not great, I treated lung cancer cell lines with TGF-beta to induce this process here. And I know you can't see very clearly, but they dramatically change morphology. They become very mesenchymal. They detach, they're not forming colonies, and they're very mesenchymal. And usually, traditionally, researchers use a couple of proteins that well describe this phenomena. If you're high in E-cadherin, you're epithelial. If you're high in vimentin and have lost E-cadherin, you're very mesenchymal. Or you can use CD44 and CD24 levels, which again, with EMT, these levels rise CD44 and the cells in this area of the plots become stem-like, which again, gives therapy-resistant traits to cancer cells. And now, if I withdraw TGF-beta, you can see the cells, within 10 days, they start becoming again morphologically epithelial, both in morphology and their protein expression of just these 4 proteins. But as I mentioned, by using CyTOF, I can interrogate all these proteins, and you can see dramatic changes with each and every time point. So you can see, like for example, E-cadherin will drop, but then start rising again once you remove the stimulus. Or vimentin will be the reverse. Vimentin is a mesenchymal marker. It goes very high, and then it can go back down once you withdraw. The plasticity and the dynamics of these processes. So now the problem is, how do I take all this information to better understand this and create this clinical tool to assess clinical specimens from lung cancer patients? One way is to use these nice fancy algorithms that help you visualize the data. So each of these dots is a cell, and then the closer the cells are to each other, it means the more similar they are to their protein expression. And then you can choose to color whatever parameter you choose. In this case, the color from blue to red is time. Here is where the cells are in high dimensional space before treatment. With treatment, within 10 days, these are where the mesenchymal cells are, and then at the red, which is after the withdrawal, and they've re-become epithelial, this is where they are. But then you can color code where the states are, the ones we define with clustering. We define 3 epithelial states, partial EMT states, which is kind of a hybrid cell. They co-express these markers. Mesenchymal cells, the M asterisk defines the stem-like cells. And then we had defined for the first time the switch from going from mesenchymal to epithelial. So there's a distinct state that the cells hadn't visited before that they have to go through to become again epithelial. Now, why is that important? Like I said, each of these states is defined by a different combination of protein expression that you could hypothetically target or assess how well the patient is doing. And then within this space, you can look at each and every protein of interest, how they are expressed in these different states that we had defined. Okay, but the goal was to build this reference map. So the same states I showed you, we defined these 8 different states. Now in this 2D map, now this is a fixed reference map that anyone can use as a tool. What you do is now, if you take a new sample that you hadn't analyzed before, analyze with CyTalk with the same markers, then you can project on here with a computational tool, and depending where each of the cells fall, it helps you decide how the patient is faring, either to therapy or with progression. And the way you do it is because we know how the proteins are expressed in each of these areas of the map, then you can define where they are in the spectrum of EMT. So for example, in the epithelial states, you have high E-cadherin, low vimentin, and then this is reversed in the mesenchymal state, right? You can see the heterogeneity and the rarity of some cells within the space, and you will only get this if you do single cell proteomics. You miss this information if you just do a Western blot or bulk RNA, right? You get all this fine-tuned information. And then I want to focus here on the MUC1 protein, which was very interesting to our study. So if you look at this area here, which is mesenchymal, you have the expected complete loss of ECAD, high vimentin, but then when they're going to they're reversing now, they're undergoing MET, you see that these are still low and high, but MUC1, which is another epithelial marker, signifies that switch. So the cells undergo a different transition to go back to where they came from. And I'm not showing it here, but we also did analysis where you can estimate the transition probabilities between the states. Why would you care about that? If I'm analyzing a tumor and cells are here, and let's say they're resistant to a tyrosine kinase inhibitor, and with treatment I see the cells fall here, I can predict what they're going to be resistant next to. That's the whole idea. Okay, so using this map, we then wanted to phenotype and analyze other tumors and liquid biopsies. So for this study, we took solid tumors from treatment-naive lung cancer patients with dissociated. We used the same antibody panel, but we also used antibodies to take out the immune, endothelial, and fibroblast elements. And then we picked what was epithelial tumor and projected on the map. And you can start to see the capabilities of this tool. So these patients that were EGFR mutated, their cells fell mostly on the epithelial and partial states. But if patients had acquired more mutations, and this has been reported, you can start seeing the tumors becoming more mesenchymal-like, which is a sign of a poor outcome, right? So you can use this to evaluate the patient samples. Now notice that this MET state is pretty much absent in the solid tumors, and that's expected. If you remember, the MET state is supposed to happen once the cells have metastasized. These are primary tumors, so it makes sense that we never saw this state here. But then we started thinking, okay, if we can't see the MET state that you could therapeutically target, would you see it then in advanced stage patients, where now there's a lot of metastases going on? So one simple way of looking into it was to take pleural effusions from late stage patients. So pleural effusion is a liquid that builds up in the lungs, and it keeps patients from breathing well. And actually, these fluids are taken out of the patients all the time, but nobody ever looks at them. Meanwhile, within this liquid, there's cancer cells and immune populations, and it could help you evaluate the patient. So we decided to take these liquid accumulation from patients and ask, do we see these phenotypes now? Do we see changes with time? Because you can take the patient's specimen with time now during treatment. And what correlations do we see with immune populations? Remember, with CyTalk, you can look at so many proteins that I can look at in detail how the cancer cells are faring and what immune populations I have in the sample. So very briefly, what we did is not only did we analyze more solid tumors, but then we took pleural effusions from 8 late-stage patients. We analyzed with CyTalk, but now we had even more antibodies to delineate what immune populations were in the pleural effusions. And then we did both characterizing the immune populations and then looking specifically in the malignant cells, what state of EMT did we detect? So for example, these are 5 new primary tumors, not pleural effusions. And when we can see all the different colors of the different immune types, and in red is the cytokeratin epithelial types. If we just take the red populations and project them on the map, we saw something that we saw before. Most of them are epithelial, right? There's our primary tumors, early stage patients. But if we, and then again, no MET state. But if we take pleural effusions from late stage patients, in some of those patients, let me go back. In some cases, we did not see any malignant cells, and that happens. But in some cases, we did find a small population of cancer cells in the pleural effusions. So when we took these populations and projected, all of a sudden, we were able to see that MET state that I talked about being important in the advanced stage. This was the first time anybody had described seeing the MET state because it's hard to study. So we saw it in the context if the patient was undergoing therapy and definitely had metastases. In the one patient that there was no metastases, there was no MET state, and in the treatment-naive patient, again, there was no MET state. And what was interesting was that we also observed a correlation between cancer cells in the pleural fluid and the existence of myeloid immune cells. There was a high correlation, which again was the first time that somebody had described this phenomena. And then another clinical aspect of using such a map, right? Here I'm showing you 2 specimens from the same patient taken 2 months apart. So the patient is undergoing combination therapy of EGFR inhibitor and chemotherapy. And then you can see that most of that patient's cells were in this state. And then after 2 months, you see the cells occupying different areas of the map. Some of them became mesenchymal, which is something that you would expect because that's what happens with drug resistance. Some of them became different epithelial, right? And you can see that in the plots, the flow cytometry plots, CyTOF. The cells in the 2 months prior patient had a lower percentage of stem-like cells compared to a specimen that was taken 2 months later. But also at the same time, there were more epithelial cells in the 2 months later acquisition of that sample. And then you can also look at a specific protein within these cells. So for example, phospho-S6 in the cancer cells we detected dramatically dropped after 2 months of therapy. And phospho-S6 is actually a readout of EGFR inhibition. So basically, and then we also saw these in vitro studies. When we took lung cancer cells and treated with a tyrosine kinase inhibitor, we saw a similar phenomenon. Some cells chose to be stem-like, some cells chose to be epithelial. And the main conclusion is that a variety of these different states, so there's such heterogeneity, could confer therapy resistance. So if you have the right tools to interrogate that, you can tailor therapy at a personalized level, one patient at a time. So what did we learn from this? You can use CyTOF to phenotype not only solid tumors, but also liquid biopsies, and we don't do this enough, especially with pleural effusions. You can use PhenoStat or variations of it, depending on the cancer type, as a translational platform and use it for a personalized approach. And then we found this distinct MET state that was first described that was characterized by high MUC1. And upon further reading, it's been shown that MUC1 confers resistance to treatments, which is what we saw in the pleural effusion. It confers resistance to inoikis, meaning it confers resistance to a programmed cell death once cells detach from the solid tumor, so it makes sense that it's important in liquid biopsies. And it's actually expressed highly in circulating tumor cells once we did further reading. So in my lab, we took that information and we said, well, if MUC1 and the MET state is important in pleural effusions, maybe it's important in circulating tumor cells as well, right? So would we see these similar proteins if now we took blood and analyzed with CyTOF? Can I detect CTCs and do further phenotyping? And then basically, can we actually use CyTOF to detect CTCs? If you know anything about CTCs, it's kind of a murky area, right? They're supposed to be low in numbers, although if you read the literature, it is believed we use the wrong markers to detect them. Many CTC studies use epithelial markers to isolate CTCs and then study them. But I just showed you how dramatically those change. So if you're just using EpCAM, which is the most used one, and it's actually FDA approved in the US, you might be losing half the cells that have undergoing EMT. So CyTOF might help us use with more markers, actually detect more circulating tumor cells. So basically, this is where we started working in my lab in small cell lung cancer. So small cell lung cancer is very different than non-small. It's way more aggressive, way more deadly, and what's interesting about it is that it has high numbers of CTCs compared to other cancer types. So it's an excellent field to go into to study this. We have PhenoStamp to study longitudinal blood specimens from patients. So again, it's very translational. Now, I want to mention that we might have to change phenotype, how we build it, with different markers because small cell is a very different cancer, but I'm just talking about the approach. And then at the same time, we can look into the immune changes. It's all about immunotherapy now, right? You can see what do the cancer cells look like, and is the patient responding to immunotherapy, all with one CyTOF run, right? And this is the collaborator and oncologist that works specifically in small cell at MD Anderson. So we've collaborated, and we take blood specimens from treatment-naive patients and patients that have relapsed to see if we can detect changes in CTCs with CyTOF. And then because I was interested in MUC1, we start doing some preliminary studies. For example, this is immunostaining of MUC1 in a primary tumor, small cell lung cancer, and the lymph node metastasis. I know the pictures are not great, but you can appreciate the increase of MUC1 expression. This is immunofluorescence staining of the same patient specimens. You see MUC1 is there in the it's an epithelial marker, but in the lymph node metastases, it's way higher, and you also have vimentin. Again, talking about that MET state that we had defined in the non-small cell lung cancer. We also saw in vitro cell line studies, these are cells that have been derived by a patient, small cell lung cancer patient, in control conditions, and when you treat with cisplatin, which is something that is used in the clinic, you see an elevated surface expression of MUC1. Again, talking a little bit about that MET state and the resistance in a liquid biopsy, because these are actually growing in suspension. So the idea was, does CyTOF work for liquid biopsies? And this is the new data from the lab, right? This is blood from a healthy donor and blood from a relapsed small cell lung cancer patient. So we basically just get 10 mils of blood, we process for CyTOF, and then we build a panel that's specific for small cell lung cancer. And you can see in the healthy donor, now CD56 is a marker, it's an immune marker, but it's also a marker for small cell. If you combine it with CD45 and it's negative, indication that this might be a small cell lung CTC. So you can see in the healthy donor, these numbers are very low, and then in a patient, you have a very high increase of these cells that might be CTCs. This is just looking at high dimensionally, all the different cell types that you can interrogate in the blood samples. So this is a healthy donor, so different colors are all these immune populations, and in black is whatever we detected as CD45 negative, CD56 positive. And in the patient, you see a way higher percentage of these cells, and then you can also start appreciating, oh look, there's higher levels of myeloid-derived suppressor cells in the patient, which is expected. Again, showing you the strength of CyTOF. Now, if we take these cells here, that with just two markers, we said it might be CTCs or not, like for example, you don't expect to have CTCs in the healthy donor, are they significantly different in other markers to help us believe this is a CTC, right? And we got great data. The black boxes, so each dot is a cell, and the black dot boxes are the levels of each of these proteins in the CD45 negative, CD56 positive cells. And you can appreciate that the cells here are significantly higher for PD-L1, which is an immune evasion marker, EpCAM, as you would expect, ECadherin, but the most interesting, MUC1. So MUC1 is not only important for, let's say, therapy resistance and survival, it can also be used as a marker to detect CTCs in the blood. And you can see that it's much higher in a patient that was relapsed after therapy compared to a treatment-naive patient. And then you can also see other proteins like SLUG as an EMT marker. And again, it's highly elevated in the patient blood compared to the normal blood. And then you can also look at immune populations. So here we're looking at T cells, right? And you can see, for example, CD8 T cells are much higher in the treatment-naive patient compared to healthy donor, and even higher in the relapsed patient. Or you can look at myeloid-derived suppressor cells, which actually help cancers survive, and you can see it's very low in the healthy donor, higher and even higher in a relapsed patient. All this from a single run of a couple of samples. So finally, the plans in my lab, what we're doing currently, and this has been just been funded by a grant for lung cancer. We're taking blood, and at the same time, we'll be taking longitudinal specimens before and during therapy, tracking all these kind of changes that I just showed you, but also enriching and growing in 3D structures so we can evaluate this CTC. Is it aggressive? Will it grow immediately? Is it immediately resistant to therapy? And evaluate, for example, if I now block MUC1, are they less aggressive? Are they less drug resistant? So then having a personalized platform per patient, and then with more interrogation with Phenostat, get a good idea of how each patient does with time and therapy. So to close the talk, I hope I gave you an idea of what cancer system biology approaches are, because that's what I was trained as a postdoc, and that's what I'm trying to bring in my lab. And cancer system biology is all about combining different expertise and collaborations and techniques. You can use not only your biological specimens, but cool technologies, and then also computational tools to get closer to personalized medicine and precision oncology. And with that, I would like to thank you for your attention, and I would like to take any questions that you may have.
There's a question there, Maria.
Hello, I'm Maria Sprodaki. I'm working in Bio1. Thank you very much for your excellent talk. I'm working with circulating tumor cells and in breast cancer and lung cancer. And I would like to ask, your technology you use, cytometry technology, is very interesting. I would like to. I saw that you use this technology in metastatic lung cancer patients and.
You mean the pleural effusions? Or
yes.
Yes.
Pleural effusion and also you have used also blood specimens or blood. Okay.
Yeah.
Have you used this technology with adjuvant patients, lung cancer patients, or breast cancer patients?
All I've showed today is lung cancer. So in the context of the non-small cell lung cancer, which was the previous study before I started my lab, it was either treatment-naive patients, solid tumors, or advanced patients that we took the pleural effusions, because as you know, it's hard to get metastatic tissue from patients that have undergone therapy, especially in at that point you don't. Now, as a postdoc, we had analyzed some breast cancer samples. Yes, but in that case, the researchers had enriched the cells and then I analyzed them. Not like I showed here that I just take the liquid and I just analyze and see what we can find. In the small cell setting, when a small cell lung cancer patient comes in, most of the time they're already advanced. That's the problem with small cell. So in that case, the only two things I have to see is either if they're treatment naive or if they already started therapy. So these are the two conditions I've used. But yeah, it's mostly lung cancer that I've done this.
Yes, but the metastatic state has a very good, there is a big muscle circulating tumor cells.
Yeah.
So it's easier to.
That's the idea. So we're trying to optimize in the small cell world, when I talked to the experts, they said there's a higher chance to also have more cells in the treatment-naive patient because they haven't been treated, so cells haven't died yet, right? So I haven't had enough samples to do the comparison if it's way more with the metastases or not, because you have to do an elaborate study. Is it the treatment that affects the numbers? Is it the metastases? If they have already metastasized, maybe they're not as circulating. There's many questions that we haven't answered yet. These are the samples.
Okay, I have two more questions.
Yeah.
The second question is if you can identify the exact number of circulating tumor cells, you can do that.
Yes, that's an excellent question that I didn't talk about. I am not selling this as an enumerating technology. We're not there yet. I haven't done the specific experiments to tell you I'm getting the exact. There's a lot of cell loss when you do CyTOF, so I can never be sure about the numbers. Now, enumerating CTCs have been shown to be important in clinical applications. What I'm offering is actually looking at the phenotype of them. So even if I can only see 30 cells, right, it's their characteristics that might be important, not so the numbers. Because the numbers don't always go with how aggressive it is, but if you can also look at, oh, these cells have this markers that we can target, or they predict therapy resistance, it gives you an additional way of utilizing CTCs. Because let's face it, most of all of us, we use CTCs as enumerating them. How high are the numbers? I'm not doing that yet. We're not there yet.
I agree with you that it's very important to
Mhm.
identify the heterogeneity of
Exactly, and that's what yeah,
and it's very important also to see the different EMT phenotypes. And,
exactly.
it's a really excellent work to identify
At this level.
Yes,
Exactly,
it's
yeah.
really fantastic. I would like also to I have a question also with the MUC1
MUC1, yeah.
marker. I think that you have tried it as an EMT marker.
Great question, because I didn't have much time to go in the details of the first part. When I built the EMT panel, I had included MUC1 because it's an epithelial marker.
Yes.
It has been mostly studied in the cancer world. It's highly elevated in colon cancer. All the epithelial tumors have it. So when I started the lung cancer project, yes, it's there in the epithelial state. It's heterogeneous though. Not all the epithelial states have high MUC1, but it's there. It dramatically drops during EMT, but then it's the first marker that goes up uniformly when they're trying to reverse. So time and single cell views is very important to understand how your protein is changing. So yes, it is an epithelial marker, but in the context of our studies, we found that although it drops during EMT, it's the marker that signifies the switch of them re-becoming epithelial.
And you have related this marker also with resistance to
Yes,
therapy?
yes, exactly. So it has multiple characteristics that is very interesting to include in our studies for the personalized medicine. Yeah.
Thank you very much.
Yeah, thank you.
You have another question?
Hello, excellent talk, thank you.
Thank you.
I'm Katie Heidi of the Cyprus Institute of Neurology and Genetics. Might be a really naive question.
There's no naive questions, yeah.
Is there a way to link, or an easy way to link the germline with this expression?
There is. I mean, I'm going to be honest, I'm a protein person and I don't have that information for this
You
stuff.
have a genetics person.
Exactly, that's what I said.
I don't know how.
Yes, the only genetic, well, I didn't do the genetics. The only thing that we've noticed with genetics in the non-small project was the association of EGFR mutation with more epithelial tumors, and then the KRAS P53, they have been shown to be more mesenchymal, and from our few specimens, we saw that at the single-cell proteomic level. But as by definition, my lab focuses mostly, like if you're completely agnostic, what heterogeneity do you see in a pure cell line? And you still see the EMT is like, there's a whole spectrum in a pure cell line. And then when you go to the patients, the only thing that we've been doing is to see, okay, now that we have the phenotype, are there driving mutations? Now, having said that, there's a lot of work that shows that certain mutations drive certain EMT phenotypes. It's just it wasn't done in the context of this study.
I was thinking also in terms of, if you want to identify if any changes are significant, can you see affecting the expressions of them?
You mean from the mutations?
No, I mean
No.
from germline.
I haven't done that work, no.
Thank you.
Yep, thank you.
Yes, we have time for Misha. Two questions. That was an excellent talk, thank you. I have a question about MAC1 as well. So we know that during the EMT process, there's a lot of epigenetic wiring that happens in cells to
Yep.
undergo that process, because
Yep.
it's too short to say that it's genetic. It could be in tumors, but in the system you're looking at, it's most likely genetic.
Epigenetic.
Right? With a TGF-beta, for instance.
Exactly, yeah.
So do you know at which stage, if you block that process, block expression of MAC1? So again,
I have no idea. That's why it's a part of my lab to figure it out. MUC1 is very interesting. It could be epigenetic, but it's also highly glycosylated. You might change everything just by blocking a glycosylation step. So we've teamed up, I haven't mentioned it here, with a person who's an expert in MUC1 to see what kind of when does it happen, how, is it post-translational, is it epigenetic? We don't know yet. All I can tell you is that it's a very important marker, in lung cancer at least, in terms of EMT and therapy resistance, and also detecting CTCs, which is, you know, something that we're trying to...
And isn't forced expression of MUC1 sufficient to...
I don't know. This is all, we went by it a very simple way. TGF-beta, I mean, I want to mention that EMT can be induced by TGF-beta, by other immune factors. It can be induced by therapy itself. So any stress that you confer to the tumor, EMT is one of the ways it uses to change its state and become resistant. So when I was a postdoc, we just did TGF-beta, and you see all these states, right? But then once you build the map, okay, now if I treat with a kinase inhibitor, where do they fall on this map? And it made sense. Maybe we should have built the map with just treating with drugs. That's there's different ways of looking at it, but yeah.